This method can help answer key questions in molecular and cellular biology field, such as identifying homologous recombination events in mouse embryonic stem cells. The main advantage of this technique is that it is accurate, reliable, and widely used.
Though this method can provide insight into identifying homologous recombination events in mouse embryonic stem cell, it can also be applied to other systems such as genetically modified cells or animals. Generally, individuals new to this method will struggle because it takes a long time and involves multiple steps.
For each gDNA, mix 30 microliters of 10x buffer for DRA1, 3 microliters of DRA1, and 10 micrograms of gDNAS per sample. Add water until the final volume is 30 microliters, and incubate at 37 degrees Celsius overnight to digest the gDNAS. Prepare a 1% agarose electrophoresis gel with ethidium bromide. Load the previously acquired samples along with a 1 kb ladder onto the gel, and run it at 30 to 40 volts overnight. The next day, soak the gel in a tray containing a 0.2 Newton hydrochloric acid solution. Use a shaker to shake the tray gently for 20 minutes at room temperature.